class. I'm happy to see you again. And today, let's prepare your notes and we already beginning to see a new chapter for cestoda species. And today, I will sharing with all of you other species in cestoda that importance in animal and also in human. So before we start, let me share my PPT materials. Okay, now we will continue in the outline and also the learning outcomes. The first one is able to mention various type of cestoda in ruminant. Sorry, I will continue share my PPT. Okay. Next, able to mention cestoda species based on taxonomy and able to mention difference in morphology and classification, and also able to mention the life cycle of the class cestoda. Next, this is the important species of Eucestoda. Last week, we already discussed and have materials about the Diplopodrium latum or the pseudopilidians and following with Echinococcus species. And today we will start study about the tiniest species of carnivore. For the tiniest species, it already exists. They have a definitive host is carnivores, maybe 7 species that importance. For the first one, Taenia hydatigena, and then Taenia taeniformis, and then Taenia multiceps, and following the Taenia cerealis, Taenia crassiceps, Taenia ovis, and the last one, Taenia pisciformis. But in the 7 species that have a definitive host is carnivore, such as canine, feline, or maybe fox, maybe wolf, yeah, that's all. They have a different characteristic. For tiny hydaticena, for the example, they have a larva stage in the muscle and the liver and the mesentery organ. And this larva stage we call Cysticercus tenuicolis. In the picture showed that this is the larva characteristic within walls layer, and the inside of the seas is consists of water, so they call it water bladder. And Cysticercus tenuicolis is the metacestoda stage. They especially find in the ships, cod, and we can observe when in the slaughterhouse and the post-mortem's infection, the existence of the Cysticercus tenuicolis. Next, Taenia taeniformis and Taenia multiceps have a different metacestoda stage. For Taenia taeniformis, the larva stage called Strobilocercus with a ribbon-like forms up to 3 centimeters long, and they have a terminal and form a bladder-like vesicle. And the four Taenia multiceps, they have intermediate host primarily is sheep, so herbivore, and the larva stage is Conurus cerebralis. Conurus cerebralis is found in the brain of the animal and also human. They can impact in neurotropics also, and commonly they find in the brain, eyes, subcutaneous tissue, and for Conurus cerebralis, we almost find in the slaughter animal for the anticipating distribution. We can use the postmortem's activity inspection. And for Taenia taeniformis, we found that the species have intermediate hosts in rats, mice, and other rodents, animals. This is the picture of the Conurus cerebralis and also Strobilucercus, the metacestoda stage of the Taenia taeniformis. Next, for Taenia cerealis, Crassiceps, and Taenia ovis, they have intermediate hosts, very different. For the first one is rodents, and then the Taenia ovis have intermediate hosts in the ships, also cots. For the larva stage, also, Cysticercus ovis found in the skeletal and cardiac muscle. You can see in this picture, this is the skeletal Cysticercus ovis. And then next, Taenia ovis in the muscle cyst, in fact, in different muscle in the slaughter shapes. And then Taenia pisciformis, this is the definitive host of dogs and other canine, and for intermediate host in rabbit. And for the larva stage, we call Cysticercus pisciformis. You can see in this picture, macro photo of the omentum region of the rodent containing large number of the pea-sized bladder-like Cysticercus of the Taenia pisciformis. And next, Taenia solium or pork tapeworm. Taenia solium have a adult stage and metacestoda stage. They are very important in zoonosis diseases because Cysticercus cellulose. The host and location for definitive host in human and small intestine, the predilect site in the intermediate host is peak. For the distribution is developing country, and for the morphological characteristic, they have for stellum and for sucker. And for transmission, human instead of raw or undercooked pork containing viable cysticercus cellulose, and the common names is pork tapeworm or pork mislis. This is the larva tapeworm in ruminant and pigs. The first one, Cysticercus bovis, and then Cysticercus tenuicolis, Cysticercus ovis, cerebralis, Conurus cerebralis, Cysticercus cellulose, and also Cysticercus for Taenia asiatica. Next, for the Taenia saginata, we call beef tapeworm or measly beef. They have two important stage. For the first one, Cysticercus bovis, we found in the cattle's muscle, and then definitive host is human. For the distribution, it's cosmopolitan worldwide, and the transmission by consuming of the raw or undercooked beef containing viable cysticercus. Next, this is the three important species of the tiny species. They can egg and transmit in human. The first one, Taenia solium. and then asiatica, and also tiny saginata. This is the part of this collection of three species. It has you can see they have four sucker and 13 hooklet for the tiny solium, but in the asiatica and saginata they only have four sucker but no hooklet. It has more or less elevated rostellum, and for the Taenia saginata, showing the same morphology of Taenia asiatica with the exception of the morphology of the rostellum, which is flat or invaginated. Next, uh, this is the differentiation and characteristics of that three species you can study by yourself. And then this species following Hymenopus nana or Vampylolepis nana. This is the rodent tapeworm, but you should take a note. For Hymenopus nana, they don't have intermediate hosts. They only have reservoir hosts that call mice, rats, and also rodents species. Next, this is the differentiation of the Hymenopus nana and Hymenopus nunta by their ductlings, colex, excised, and also intermediate hosts. And for the Hymenopus diminuta, they have intermediate hosts. For the reservoir has none, yeah, only intermediate hosts. And for the age, you can see the morphology of the age. Hymenopus dimunta and Hymenopus nana is very different. The eggs of Hymenopus dimunta round and slightly oval with size of approximately 17 and 18 micrometers with a thick serrated outer membrane and a thin inner membrane. And the oncosphere has 6 central hooks. And for the Hymenopus nana, they have an oval or subspherical with a small size, and the inner membrane has two pores from with a polar filament span out between the membrane, and the oncosphere also have a 6 central hooks. Next, Diplodium caninum or double pore tapeworm or cacing pita anjing in bahasa, but you can identify the Diplodium caninum as dog tapeworm, right? For the definitive host, by their name is dog tapeworm, so the definitive host is dogs, cats, carnivore, and occasionally in humans. So this species is zoonosis disease. An intermediate host is Stenocephalides canis and also Stenocephalides fellis. And for limitation, I know the intermediate host is ectoparasite, so they have metacestoda stage called cysticercoid. And next, the eggs form eggs package or egg ball containing multiple oncosphere with 3 pair of hooklets, right? And this is the double pore in the left side and the right side. Next, prepatent period and environment. You can see this is these species have 2 to 3 weeks following infection. They have shading proglottid in the feces. And then this is the morphology. And then this is the Diplodium caninum life cycle. The graphic proglottid are passed, in fact, in the phases of emergence from perianal regions of either animal or human host. Following the proglottid disintegrated and released the egg package. And then larva of the typical flea intermediate host ingested its pocket and oncosphere hatch and develop into cysticercoid. Cysticercoid after then ingested by flea larva mature in the adult flea, which continue to harbor infective. And the definitive host is infected by ingested fleas containing cysticercoid. And then the incidental human transmission via ingested of the infected flea, they will becoming infected in the small intestine, and cats and dogs can harbor fleas infections to human also. Next species is Dipilobothrium or Spirometra mansoni. The definitive host is dogs and cats, and intermediate host is cyclops, and following with frogs and snakes. This is the Sparganomansoni life cycle. You can see they have embryonate eggs following in water. We call Coracidia H. And then ingested by crustacean or capepod and developed by procercoid, and then following with plerocercoid. And plerocercoid is metacestoda stage found in the frog and or snakes. Next species, the member of family Daphnia, we can study three important species. The first one, the Phallinia progliatina, following with Rhaetina tetragona, Rhaetina chastinochilus, and Rhaetina echinobotrida. This one. This is the infection appears when the large and small intestine infected with the Rhaetina species. And then this is the comparative morphology of Rhaetina found in domestic chickens. And then this is the picture. So the Rhaetina tetragona, and the morphological characteristic, the adult length up to about 25 centimeters, with the suckers oval. The sucker is oval. The rostellum arm 8 until 10th row of hooks, with the egg capsula is capsule containing 6 until 12 each. And then Rhaetina echinobotrida, they have length 25. Hooks are present on the pot of rostellum and also sucker. And then the graphite segment containing eggs pocket within capsular. This is the Rhaetina chasticillus, right? And this is the average length. The rostellum is broad with 400 and then 1500 hooks. And then next, genus Monasia. Genus Monasia, also called adult tapeworm in Ruminantia. We study two species, Monasia expansa and Monasia benedini. They have intermediate host is cercoid in forage mite from the family Oribatidae, found in ruminants. This collection is unarmed with only 4 sucker. For Monasia expansa and benedini has interploglottal gland, arranged transversal along the middle of the body, irregular triangular eggs, we call piriform apparatus, like this one. And this one also. This is the life cycle of Monasia when the definitive host is infected when it eats an infected intermediate host containing metacestoda stage is a cysticercoid in the body of Oripide mites. And this is the last slide for description segment nature of Monasia benedini and also Monasia expansa. For the interprognathid gland are arranged short and compact in the center of the segment of Monasia benedini, segment prognathid, and then for Monasia expansa in the interprognathid gland are arranged longitudinal of the posterior segment. And this one is the quotes of the day from me. And that's all. our presentation, my presentation, and we already finished study about the cestoda species completely. So thank you guys and see you in the another other materials.